Welcome to BCM201. Today we are going to look at the use of polymerase chain reaction in science, a technological development in science that has revolutionized molecular biology and other biological sciences that work with DNA. PCR, or polymerase chain reaction, allows us to identify and amplify rare DNA sequences directly out of complex DNA mixtures. This discussion will give you an overview of the purpose of PCR, the history and development of the technique, what PCR is used for, how PCR works with a focus on the process at a molecular level, current applications of PCR, advantages of PCR, shortcomings or disadvantages of PCR, and to finish off, the potential future applications or extensions of the PCR technique. So, what does the PCR technique allow us to do? PCR enables the synthesis of specific DNA fragments using a DNA polymerase enzyme, which takes part in the replication of the cellular genetic material. The DNA polymerase enzyme is an enzyme that creates DNA by joining the nucleotides together. This enzyme creates a complementary sequence of DNA as a small fragment. A primer is then connected to one of the DNA strands in the specific site chosen to start the synthesis. A primer is an oligonucleotide that acts as the leader for the DNA extension. These primers limit the sequence to be replicated, and the result is the amplification of a particular DNA sequence with billions of copies. So let's talk about how all this started. In the spring of 1983, Dr. Kary Mullis, you can see his image in the right-hand corner of the slide, while trying to find a way to increase the demand for oligonucleotides, which, before automation, were made by hand in laboratories, and trying to save his colleagues' jobs from a machine who could do this task in a couple of hours, as well as create a simple lab test that could be completed in a hospital laboratory in one shift to help identify genetic mutations. Dr. Kary Mullis began hypothesizing ways to make this happen. In his paper, Specific Enzymatic Amplification of DNA in Vitro, the Polymerase Chain Reaction, Dr. Kary Mullis and colleagues relayed to the world how the PCR technique would work. There was still, however, one minor problem. After the first round of strand copying, the DNA had to be heated to almost boiling point to denature the newly formed double-stranded DNA, which allowed the strands to separate, providing a new single-strand template for the next round of amplification. The original method used the clean-out fragment of the Escherichia coli DNA polymerase I, and its inability to withstand heat caused the original PCR technique to be slow and laborious. Heating the clean-out fragment inactivated the polymerase, so more enzyme was required at the start of each cycle. So how did they fix the problem with the clean-out fragment? Let's go back another decade to 1976. Although it wasn't known at the time of discovery, Chen and colleagues isolated what would become the most famous representative enzyme among the thermostable polymerases, DNA polymerase I, isolated from the bacterium Thermus aquaticus, known as TAC. This enzyme had the ability to withstand the protein denaturing conditions that were required during PCR cycling without the need to refresh DNA polymerase time and time again. It was the use of this enzyme that could withstand the temperature required in the repeated denaturation steps that led to the success of the PCR technique. In 1993, the Nobel Prize in Chemistry for contributions to the development of methods within DNA-based chemistry was awarded jointly with one half to Kary B. Mullis for invention of the PCR method. PCR is used over a multitude of disciplines. To name a few: food sciences, forensics, the Human Genome Project, and disease diagnosis. PCR is being used more commonly in the food industry as a method to help guarantee the quality and safety of food. PCR is currently used by clinicians and scientists to diagnose disease, clone and sequence genes, and to carry out quantitative and genomic studies using a fast, sensitive method. The technique is also used in forensic medicine to identify criminals. PCR has been used to analyze the bacterial DNA of ancient mummies, which helps to identify the occurrence and frequency of infectious disease in historic populations, and may also provide evidence of the evolution of microorganisms and their associated diseases. One of the most significant uses of PCR to date was its use in the Human Genome Project, a project that analyzed the complete DNA sequence, mapping all genes of the human genome.
So components chemically necessary for PCR. First of all, we need our DNA template. So this is our target sequence we're going to use for our initial template that we want to copy. We need DNA polymerase enzymes to synthesize the new DNA, replicating the target DNA that we've chosen. We need primers that to bind to each end of our target sequence, and nucleotides. We need all 4-deoxynucleotide triphosphates, and we're going to talk about a couple of these more extensively on our next slide. So primers are short synthetic oligonucleotides that are complementary to the DNA sample that we want to amplify. So the primers are a starting point for our DNA polymerase to build upon, and primers help limit the amount of DNA to be replicated so that we're getting a small sample and making a lot of copies of it. So the DNA polymerase is an enzyme which is able to synthesize DNA fragments complementary to our target that we've selected. DNA polymerases that are used need to be heat resistant and synthesize the DNA using the provided template. Common DNA polymerases that are used are Taq DNA polymerase, which is from a bacterial species called Thermus aquaticus, and we use this because it's very thermostable. And another one that's commonly used, not as thermostable, but it's called PFU DNA polymerase, and it's from an archaean microbe called Pyrococcus furiosus, and we use this due to its high fidelity, which means that we have not many errors when we build our new DNA. So polymerase chain reaction, how does it work? So what actually happens for scientists to get this reaction to occur is all of these components are mixed into test tube or an appropriate vessel, and it's placed into what we call a thermal cycler. And this thermal cycler precisely heats and cools and heats the sample to facilitate the process of polymerase chain reaction to actually occur. So all those things go into the thermal cycler. So there's actual processes that occur inside this thermocycler, and those processes are denaturation, annealing, and extension. And what we can say is that all these are the amplification of our initial DNA template, and this cycle, the cycle of the 3 steps, is repeated again and again, maybe 20 to 40 times. And each time this cycle of PCR is repeated, it exponentially amplifies the number of DNA strands. So the first strand becomes two strands, and then those 2 strands become 4 strands, and then 4 becomes 16, and so on and so forth, until after 30 cycles, which can happen in a couple of hours, there are over 1 billion strands of that initial DNA sequence. But let's talk about these processes in a little bit closer. So our first step of our PCR is denaturation. So if we look over here, we have our parent DNA. This DNA is heated to 95 degrees Celsius to separate the 2 strands. So you can see here that we now have 2 separated strands of our DNA. And that brings us to our next process, which is called annealing. So those 2 DNA strands are cooled down to 55 degrees Celsius, and then the primers are bound to the separate strands of the DNA on the opposite ends. So we can see we've got a primer here attached, and down at the other end we have a primer attached. And as we said before, these primers are complementary to our DNA template. and then we're going to get our nucleotides are then going to come down in extension. So the DNA polymerase is an enzyme that activates the nucleotides to come along and attach themselves in the right order to our initial DNA template and builds up the 2 strands, and therefore we get 2 more copies of the DNA strand. So if we get to here, we've got step 4, or step 4 covers the whole process. Amplification, and we can see along here we have our 2 new DNA strands making 2 whole strands, and then we take the process to continue on further. So we repeat this cycle again and again, as we said before, 20 to 40 times. Each cycle of PCR exponentially amplifies the number of DNA strands 2 to 4 to 16, and so on and so on and so on, and this can be done over a few hours. And again, like we said, after 30 cycles there are over 1 billion strands of this initial DNA sequence. So current applications of polymerase chain reaction. So PCR is a really useful tool in the study of genetic diseases such as haemophilia, cystic fibrosis, muscular dystrophy, Huntington's disease, sickle cell anemia, just to name a few. Obviously, there are so many things that are genetic diseases and things that this can be used to investigate further. It's great in the detection of pathogens, whether they be viral, bacterial or fungal. PCR is fast and specific, and it removes the need to do long culture samples. We can also use it in forensic medicine and forensic science for identification, whether that be the study of the past or even for criminal investigations. Another really interesting one is disease identification in dentistry. PCR can be used to help identify potential or rapid detection of periodontal diseases, dental caries, endodontic infections, and other oral cancers. Specific examples of the application of PCR. So PCR testing is really relevant to myself, Carly, in my everyday life. My husband and I own and operate Gazander Oysters in Coffin Bay in South Australia. Recently in 2016, the Tasmanian Pacific oyster industry has been devastated by the appearance of the POMS virus, also known as OSHV1, which is a herpes virus. POMS basically kills 95% of the stock on your farm very quickly without any warning. Prior to that, in 2010, it also wiped out most oyster farms in the New South Wales estuarine growing areas. The virus was first discovered in France and has had devastating effects in many countries so far. PCR testing is used to test for the OHSHV1 virus. Also, PCR was used to sequence the genome for this virus. So South Australia still remains disease-free, and there's an importation ban into the state of any live Pacific oyster spat or any other size Pacific oysters, and any equipment used in any infected waterways is in place. So basically, we've been unable to buy any spat from Tasmania, which is really important to our business. PIRSA and the SA oyster growers employ active surveillance for POMS outbreaks through biannually testing of spat samples and passive surveillance through testing of oysters with suspicious mortality events using PCR. So newly opened South Australian hatcheries use PCR in testing of oyster family lines to prove oysters resistant to the OSHV1 virus and to prove maintenance of a POMS-free record of health for their hatcheries. So PCR plays a really important role in protecting and monitoring of Australian waters in regards to the POMS virus, and it is a tool that my business relies on every day to maintain our disease-free environment. So I thought that was a really relevant example of how it is a beneficial discovery and use in an everyday situation.
Advantages of PCR. There are different advantages in PCR, and first I'm going to talk about the ones in medicine. PCR has been used to identify infectious diseases and has assisted with the diagnosis of genetic disorders and cancers. PCR is able to detect cancer before symptoms have occurred, which therefore suggests that there would be a higher chance of being cured. In mainstream medical diagnosis of cancer, usually it is picked up once symptoms have occurred, and that therefore makes it harder to treat. Infectious diseases are hard to identify, although PCR produces a rapid identification process for these illnesses. PCR can detect fungal, bacterial and viral pathogens through the identification in DNA. Epidemiologists use PCR-enhanced DNA samples from human remains to trace the evolution of human pathogenic viruses. This is beneficial as it assists with finding the main source where the virus was found. For example, if there is an outbreak of a particular disease, it can be tracked back to where it originated. Advantages in biotechnology. Gene cloning has been developed for biotechnological purposes and has been beneficial for producing important proteins as medicines. This technique is highly sensitive, which means that it can detect and amplify just one DNA molecule in almost any type of sample, including ancient ones. PCR has allowed for successful cloning of rare undegraded DNA segments isolated from samples more than 40,000 years ago. This has been beneficial in tracing ancient human migrations and has been beneficial for historical research. Shortcomings of PCR. PCR has been an extremely beneficial advancement in science, although there are some serious limitations with this technique. I'm going to speak about these below. PCR is highly sensitive, meaning that even the smallest bit of contamination of a sample of DNA can lead to misleading results. To overcome this, it is important to understand the information about the DNA sequence being replicated. The DNA polymerase in the PCR is prone to errors and can lead to mutations in the fragment generated. Designing primers to generate a PCR product requires some prior sequence information to ensure its accuracy. What is the future for PCR? It is suggested that PCR is promising for greater insight into future gene combinations. PCR is continuously being explored and new techniques or advances to the conventional PCR are constantly being developed. I'm going to talk about some of the extensions below. Quantitative PCR and real-time PCR. This assists with PCR when there are higher amounts of DNA present in a sample. For example, if genes are amplified in tumour cells, it can reveal the increased sequence as a result. Reverse transcriptase. This type of PCR has a beneficial role in the future as it enables PCR to detect sequences from living cells, as opposed to dead tissues. Digital PCR. Digital PCR was developed as a further extension to the conventional PCR as it enables more accurate, sensitive and reproducible targets. This is continuously being explored as there is a promising future for this technique with more research to be done. It is a cost-effective technique which is beneficial, although it needs some small changes to make the small reaction volume and high sensitivity more precise. This technique is promising for the future of PCR as researchers are looking into a more cost-efficient method. Some of the improvements needed for PCR. Some of these include more cost-efficient, less sensitive and more accuracy. There are a few different things where PCR may be able to help in the future, and these include diagnosing cancer earlier than in medical practices, preventing spreading of infectious and pathogenic diseases, and gene cloning. As you can see, there are many different advantages and limitations to the technique, although the benefits of this technique outweigh the few limitations. In the future, this will be a successful method in a large variety of different issues. These are our references, and thank you for watching our presentation.